Hello and welcome to my YouTube channel, AnatomyMentor. Hope you're all doing well. Today I'm going to discuss a topic which is in connection with the different lectures of neuroanatomy. This is the histology of the cerebral cortex, which we shall be talking about today. So starting with today's discussion, our learning objectives are that we are going to enumerate the layers of the cerebral cortex and we shall enlist different cell types of these layers. Also, I'll explain the histological features and connections of the various cell types within the cerebral cortex and apply the knowledge of anatomy, histology to explain the clinical condition which is known as the Alzheimer's disease. So these are our learning objectives for today. And starting with today's topic, you know the cerebrum, it has an outer covering of gray matter. And that outer layer of gray matter, which is surrounding the cerebral hemispheres, is known as the cerebral cortex. And this cerebral cortex, it is folded upon itself to form the gyri and the sulci. And these are actually the convolutions of the cerebral cortex, and they serve the purpose of increasing the surface area available for function. Okay, and also you should know that inside this gray matter is the white matter or the medulla, which conveys fibers between different parts of the cortex to and from other parts of the central nervous system. And then further deeper within that white matter are certain collections of gray matter, which are known as the basal nuclei. So this is the general organization of gray and white matter in the cerebrum. Now, we know that the cerebral cortex is gray matter. Its area is about 2.5 square feet. And remember, only 1/3 of this is found on the free surface, which means the rest of it is present within the troughs or the grooves, also known as the sulci. And the number of neurons in the cerebral cortex are about 14 billion. And the thickness of the cerebral cortex is varying from a range of 1.5 to 4.5 millimeters. The thickest is going to be over the crest of a convolution, whereas the thinnest is going to be within the depth of a sulcus. Now, the important thing to keep in mind when we're discussing the histology of the cerebral cortex is that it does not have a uniform structure. And this is the basis of classification of the cerebral cortex. So how can we classify cerebral cortex? Well, the classification, which is based on the evolutionary development, that is known as the phylogenetic classification. And based on this classification, the neocortex, the cortex will be either neocortex or it will be allocortex. First, let's talk about the neocortex. Neo, as the name shows, is the most recent in evolutionary development, and it forms 90% of the surface of the cerebral hemispheres. So it is also known as neopallium or the isocortex. It has the characteristic 6-layered arrangement of cells. Now, the remainder of the cortex is known as the allocortex, which means the other type of cortex. And this is the one which consists of 3 to 5 layers of cells, and it includes the paleopallium, that is the olfactory cortex, and the archipallium. Archi means this would be the oldest, considering the evolutionary development, the most primitive. This is the one which is present in the areas of hippocampus and dentate gyrus, which make a part of the limbic system. So that is the phylogenetic classification of cerebral cortex. Now keep in mind the functional units of the cerebral cortex are disposed vertically corresponding to the general orientation of axons and dendrites. So here we have a histomicrograph of the cerebral cortex. On the left, you can see is the one which is stained with methylene blue, which is a special stain. Whereas on the right, the histomicrograph has been showing us the hematoxylin and eosine stain. And if we look over here, these are the layers of the cerebral cortex, which are represented by Roman numerals. And from the superficial aspect over here, if we go down to the deeper aspect, these layers are numbered from 1, 2, 3, 4, 5 to 6. So these are the 6 layers, and this layered structure can be slightly appreciated on a lower magnification. It is because of the characteristic neurons which are present within each layer, and we shall be talking about these in the next slides. So here are the 5 characteristic types of neurons which are present within our cerebral cortex. These are pyramidal cells, the granule, also known as the stellate cells. The word stellate literally means star-shaped. Also, we have the fusiform cells. The word fusiform literally means spindle-shaped. The cells of Martinotti and the horizontal cells of Kagel. All of these cell types will be present in cerebral cortex, but most numerous, remember, are the pyramidal and the stellate cells. Now, here are the layers of the cerebral cortex, the characteristic 6 layers from the superficial to deeper aspects, layer number 1, 2, 3, 4, 5, and 6. And if we look at these layers, we can see the most superficial is known as the molecular or the plexiform layer. It has transverse fibers and some scattered neurons. Then we come over to the external granular layer over here, lying beneath the molecular layer. It has mainly a large number of the granule or stellate cells. Then comes the pyramidal layer, which is layer number 3. It has these pyramid shape, or you could say these are like triangular or flask-shaped neurons. And a smaller number of stellate and basket cells will also be present. Then comes the layer number 4, which is internal granular layer. Again, it has a higher concentration of the granule or stellate cells. Then the layer number 5, which is again a pyramidal layer. It is also known as the ganglionic layer. It has giant pyramidal cells. And the innermost layer, that is the deepest layer of cerebral cortex, this is the polymorphic or multiform layer. The name is self-explanatory. It shows that the neurons over here have different shapes and have different sizes. The layer number 4 and 5 also has, you can see, these transversely arranged fibers. So they're also known as the outer and inner bands of Baillarger, respectively. Okay, so now let's look at the nerve fibers. How are they arranged within the cerebral cortex? Well, there are 2 types, the radially arranged fibers and then those which are tangentially arranged. The tangential fibers are most concentrated in layers number 4 and 5, which I just showed you, where they're known as the outer and inner bands of Baillarger, respectively. Now, the bands of Baillarger are particularly well developed in the sensory cortex, because over here there is a higher concentration of the terminal parts of thalamocortical fibers. Now, there is a certain area of the cerebral cortex in the occipital lobe, that is the visual cortex, where the outer band of Baillarger is so thick that it can be seen with the naked eye, and therefore this is known as the stria of Jenner. And the visual cortex in the walls of the calcarine sulcus is known as the striate cortex. So that is a point to remember, to keep in mind whenever we're discussing the histology of the cerebral cortex. Now we're going to discuss these cells, the different types of neuronal cells that are present in the cerebral cortex one by one. Let's first talk about the pyramidal cells. So here you can see the pyramidal cell, which is the most abundant of the cortical neurons. It is triangular shaped and its size is about 10 to 50 microns. And in case of the very larger ones, the largest sized pyramidal cells, they have a special name. They're known as the BET cells. They can be up to 100 microns in size, and they are located in the precentral gyrus. You can see over here that there is a dendrite which projects upwards from the apical region of the pyramid-shaped cell towards the pale surface. And there are also numerous basal dendrites projecting horizontally from the cell body, whereas the axon, it emerges from the base of the pyramidal cell and enters deeper layers of cerebral cortex, and also it's going to enter into the white matter. Now, the other cell type which is abundant is the stellate or granule cells. Like I told you before, the word stellate literally means star-shaped cells. So these are the small size cells, and they have about 8 micron is their size, and they have multiple branching dendrites, and they have a very short axon. Now, the other cell type in cerebral cortex are the fusiform cells. Fusiform literally meaning these spindle-shaped cells. So you have an oval cell body, and their long axis is directed at right angles to the surface of the cerebral cortex. The axon arises from one side of the cell body and it ascends superficially, whereas the dendrites extend from both sides and they're going to go into the superficial layers as well as into the deeper layers. Then the cells of Marti-Notai, you can see they're present throughout all the levels of the cortex, and they're small polygonal cells if you look at the cell body with a few short dendrites, but the axon extends towards the surface and eventually it is going to, you can see, bifurcate, that is split into two, usually in the most superficial layer. Then the last cell type, that is the horizontal cell of Gajal. You can see that these cells, they are found only in the most superficial layers, and they are small and they're spindle shaped. They're oriented parallel to the surface of cerebral cortex. And the dendrites, they emerge from each end of the cell, while the axon, it is going to be parallel to the surface of the cortex, making contact with the dendrites of the pyramidal cells. Okay, so over here now we have this summary of the different cell types which are present within the cerebral cortex. And we can see here are the layers written in Roman numerals: 1, 2, 3, 4, 5, and 6. So this is from the superficial to the deeper aspect. These are the 6 layers of the cerebral cortex. And we can see over here that in the layer number 2 and 4, we find mainly the stellate cells, which are the small star-shaped cells. And if we look over here at layers 3 and 5, we can see they have these pyramidal cells, and they are largest in the 5th layer, where they're known as the bed cells. And let's look over here at the superficial layer, which has these horizontal cells. And this here is the deepest layer, layer number 6, which is the multiform layer because it has neuronal cells of different sizes and shapes. Let's note over here that the afferents, those nerve fibers which are entering cerebral cortex, they're going to move upwards and they're going to synapse in the cortex with the dendrites of the neurons. Now, what about the efferents? That is, this nerve fibers which are leaving the cerebral cortex. They are typically the axons of the pyramidal cells. And as you can see, they are emerging from the base of the pyramidal cells. So this is a general organization. Over here, you can see also the fusiform cell in the 5th layer. You can see it is directed, the cell body is at right angles to the surface, and its axon is also projecting upwards. So this is a general organization of the different neuronal cells in the different layers of cerebral cortex. And over here you can see that this here is then again a summary. This is a silver impregnation histomicrograph, which is showing us the layers of cerebral cortex. And superficially, you can see the pia mater, which is covering the surface of cerebral cortex. And beneath it, you can see these characteristic 6 layers, which have been labeled over here as the most superficial is the molecular layer. Then comes external granular, external pyramidal, internal granular, internal pyramidal, and the 6th one is the multiform layer. And you can see that the pyramidal cells are more in number, and they're prominent within the 3rd and 5th layers, whereas the stellate or granule cells are more prominent in the 2nd and 4th layers. Okay, so now moving forwards, you have to remember, other than the neuronal cells, which are the functional cells, there are also some supporting cells within cerebral cortex, and these are astrocytes, oligodendrocytes, and microglia. So in this histomicrograph, you can see that these are the pyramidal cells of cerebral cortex, but also present amongst them are the astrocytes. And with this stain, with this special stain, which is the silver stain, we can see the pyramidal cells with much better clarity. We can see their cell bodies, and we can also see the dendrites and the axons. You can see these axons which are emerging from the cells. And here A is representing the astrocytes, which are the supporting cells that are also present within the cerebral cortex other than the neurons. Now, what is the histological classification of cerebral cortex? First, we talked about the phylogenetic classification. Now, let's come to the histological classification. There are 5 regional variations described in the structure of the neocortex. And these can further be divided into 2 groups. The first group is known as the homotypical variants. In the region where there are homotypical variants, remember all the 6 layers are present. And these are the regions of frontal lobe, parietal lobe, and the poles of the cerebral hemispheres. The second group is the region which contains heterotypical variants. Now, heterotypical means that the characteristic 6 layers are not going to be present in their typical form, but a few layers will be predominating. So this can be either of 2 types. It can be the granular cortex, in which layers 2 and 4 are well developed, or it can be the agranular cortex, in which layers 3 and 5 are well developed. So based on this classification, when we're talking about heterotypical variants, first we're going to discuss the granular cortex. So over here, like I mentioned, the layers 2 and 4 are well developed, and they are the parts which contain the densely packed granule cells, also known as the stellate cells. And over here, the layers 3 and 5, the ones which contain the pyramidal neurons, are poorly developed. So layers 2 through 5 merge into a single layer of predominantly granular cells. And this type of cortex is found in the postcentral gyrus, also in the superior temporal gyrus, and in parts of hippocampal gyrus. So it is mainly the sensory cortex which contains the granular cells. Now, the other heterotypical variant, which is the agranular cortex, is the one in which the granule cell layers 2 and 4 are practically absent, but the pyramidal cells in layers 3 and 5 are densely packed and very large. This type of cortex is present in the precentral gyrus, that is the motor area, and also other parts of frontal lobe. So the motor cortex is mainly the agranular cortex. Now that we have discussed the histology, the basic histology of cerebral cortex, we're going to discuss a clinical condition which is quite common now in the world, and we're going to discuss it at the histological level as well. So we're talking about dementias. What are dementias? These are medical conditions in which there is progressive, generalized deterioration in brain function, especially in the higher cortical functions. And there are many different forms of dementia with varying genetic components, but the most common form of dementia in the world is Alzheimer's dementia. Now, what are the stats when we talk about Alzheimer's disease? Remember, right now, about more than 55 million people worldwide have Alzheimer's disease, and women are affected more than men. And this can occur after 45 years of age, but the incidence particularly increases after 65 years of age. So the elderly population is vulnerable to Alzheimer's dementia. Remember, it affects both neuronal cell bodies as well as the synapses within the cerebrum. The typical symptoms of Alzheimer's disease in the patient are increased memory loss and confusion. Thinking and reasoning skills are also affected. There are also personality and behavior changes and language problems. What about the histopathology of Alzheimer's dementia? So in this condition, there are 2 typical features, histopathological features. One are the neurofibrillary tangles, and the other are the neuritic plaques. So first, let's look at these tangles over here. This is a stain section of the cerebral cortex, and you can see over here there are neurofibrillary tangles of tau protein in association with microtubules. These are the primary biomarkers of Alzheimer's disease. Also, you can see the plaques, which are also known as senile plaques or neuritic plaques. These are extracellular deposits of the protein, which is known as beta amyloid protein. So these are the characteristic histopathological features which are diagnostic of Alzheimer's disease. So this concludes with today's discussion on the histology of the cerebral cortex, in which we have discussed first the phylogenetic classification and then the histological classification of cerebral cortex, and looked into a little detail of the size and shapes of the different neuronal cell types that are present in cerebral cortex and the layers of these cells, their arrangement in cerebral cortex. I hope you found today's discussion interesting and it was easy for you to understand. If you found it helpful in understanding the histology of cerebral cortex, then don't forget to like and subscribe to my YouTube channel, Anatomy Mentor. You can do that quite easily by just clicking on my picture, which is going to appear at the end of today's video. And also don't forget to hit the bell icon for regular notifications of my educational videos and my presentations. So take care and stay tuned, as I will be coming again with more topics of histology and also embryology and neuroanatomy. Thank you and take care.